Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullah. Our lecture for today is going to be about pathogenesis of viral infections. So to start with, we need to familiarize ourselves with certain terminology when we're talking about viral epidemiology. The first term is endemic, epidemic, and pandemic. These three terminology come together when we talk about the spread of virus infection. So when we say endemic, the disease is present at fairly low but constant level at a certain geographical location at a certain point of time. So when we say like HIV is endemic, it's present at fairly constant level, does not necessarily have to be low or high. It could be high, such as certain villages or even countries in Africa where HIV is present there at fairly constant level. Endemic معناته مستوطن. Epidemic is an infection with greater than usually found in a population. Epidemic. So the infection is greater than usually found in a population at a certain point of time and at a certain geographical location. And the good example of epidemic is the influenza virus during the winter month. So the number of people who becomes infected with influenza virus during winter months is high in Jordan. This is an example of an epidemic. Pandemic, infections that are spread worldwide at a certain point of time. And a very good example of pandemic infection is the COVID-19 or the SARS-CoV-2, which causes the COVID-19, the corona virus, which we're living at today. So it is a pandemic. Infectivity is the frequency with which an infection is transmitted when contact between a virus and the host occurs. The frequency with which an infection is transmitted when contact between a virus and the host occurs. So an example of the infectivity is once again, we can talk about the coronavirus, the SARS-CoV-2. How infectious is it? It is highly infectious, especially in limited space and with poor ventilation. It is highly infective. Disease index, the number of persons who develop disease divided by the total number of who are infected. So this is called disease index. It is easy to find the number of persons who develop disease, but the total infected, it is hard to tell. Once again, if we want to talk about the coronavirus, the SARS-CoV-2, the number of persons who develop disease are the numbers that we're living today. So up to today, maybe we're having 66 million persons who are diagnosed and who have disease. And the number of totally infected, it's hard to tell. It needs serological studies to see seroconversion among these individuals. And if there is seroconversion, and what do we mean by seroconversion? Seroconversion is the state of presence of antibodies against specific virus, even if that person did not develop disease. We know that in the coronavirus case, that certain persons, they become infected, but they do not develop the disease. The only way to tell is to do the serological study and look for the IgG in their blood. So this is the number of persons who develop the disease and have the symptoms divided by the total infected. Virulence is the number of fatal cases divided by the total number of cases. Virulence, the fatal cases. And a good example, once again, is the coronavirus. The number of fatal cases, we're talking about 1,600,000, something like that, divided by the total number of cases. And this is the virulence of the infection. When we talk about the incidence and prevalence, those two terms come together. And the incidence is the number of new cases within a specific period of time. And usually it is represented as a percentage. So when we say the incidence of the coronavirus in Jordan, if we report today, let's say 3,000 cases of coronavirus, then we're going to divide them by the total number of cases, which are almost around 300,000. So the incidence is 1%. So it's the number of new cases within a specific period of time divided by the total number of cases. Prevalence is the number of cases of a disease that are present in a particular population at a given time. So the prevalence is the total number of cases which are present. The total number of cases which are present. Once again, go back to the incidence. It can be given as an actual figure, or it can be given as a percentage. What does a pathogen have to do? So pathogens or viruses, what do? They infect the host cell. They reproduce or replicate themselves and then spread to infect nearby cells and ensure that its progeny are transmitted to another host. So it's going to spread to nearby cells, and it can also spread to other humans or even infect other species, especially when we're talking about animals. When we're talking about viruses which can infect humans, animals, birds, and a good example is the influenza virus. So what are the routes of entry or the routes of infection of viruses in humans? We have the first horizontal, we have the second vertical, and we have the third one, which is zoonotic. Horizontal transmission is person-to-person transmission, and this can occur via three routes: either inhalation through the respiratory tract, ingestion through the GI tract, or inoculation, which is entry through wounds or skin abrasions, or entry through mucous membranes, transfusions, injections. So let's start with the first, inhalation. Inhalation occurs via the respiratory tract via respiratory droplets or aerosols. Respiratory droplets or aerosols, and good example is the coronavirus, SARS-CoV-2, respiratory syncytial virus, mumps, measles, rubella, varicella zoster virus, rhinovirus, coronavirus. Rhino and coronaviruses are viruses which are commonly seen during the winter months, and they usually cause the common cold, الرشوحات العادية. Ingestion via the gastrointestinal tract, and another way of saying it is the fecal oral route. So viruses are being shed into the feces, and if these contaminated feces with the viruses can get into the water resources, or fruits and vegetables are being irrigated with these contaminated waters, and the humans come and ingest them, they can get infected with these viruses, such as the hepatitis A virus, the rotavirus, the astrovirus, the calicivirus viruses. The third type of the horizontal route is inoculation, as we said, through skin abrasions, mucous membranes, transfusion, injection, and also through shared syringes in drug abuse or transplants. The second is the vertical transmission from mother to fetus. And during the vertical transmission, we have three phases of vertical transmission. We have the transplacental, which is during pregnancy, we have during delivery, and we have after delivery during breastfeeding. And examples of transplacental infections or transmission that can occur is the cytomegalovirus, rubella virus, and HIV virus. During delivery, hepatitis B, hepatitis C, herpes simplex virus, HIV, and human papillomavirus can be transmitted during delivery. Among all these, herpes simplex virus is an absolute no-no of normal vaginal delivery. So if pregnant woman is diagnosed with having herpes simplex virus, vaginal herpes simplex virus, then she should not give birth via normal vaginal delivery. She should rather give birth through cesarean section. Breastfeeding, we have example of cytomegalovirus, hepatitis B virus, and HIV virus. And if we look, we can see that HIV virus can be transmitted via all of these vertical stages. Zoonotic virus infection, which occurs from animals to humans. We have animal bite, example, rabies اللي هو داء الكلب, which can be transmitted to humans from rabid dogs, الكلاب المسعورة. Also, we have the insect bites, such as the dengue virus and the West Nile virus, and these viruses are transmitted through bites of the mosquitoes. Also, we have animal excreta, example, the hantavirus and the arenavirus, and in this case, we have mice which are infected with the virus, and then the virus is going to be excreted in the feces of these viruses. The feces of these viruses are going to become dry, and they might become airborne, and then humans might inhale them and get infected with these viruses. So once again, the sites of virus entry, they can enter through the mucous membranes, such as in the eyes, in the mouth, also through the respiratory tract. In the respiratory tract, we have protective mechanisms such as the cilia, and we have the mucus secretions, and we have also the lower temperature within the respiratory tract, especially within the nasal cavity. It's around 33 to 34. Also, we have the IgA immunoglobulin, which can neutralize any invading virus. Also, we have the alimentary tract or the GI tract, and in the GI tract we have the gastric acid and the bile salts, which have sterilizing effects. Also, the skin is one of the non-specific defense mechanisms of the body, and if there is an abrasion, if there is a cut wound, then viruses might enter through this cut wound, or if the virus is transmitted through an arthropod vector like a mosquito can bite and introduce the virus inside the body. Also, we have through the urogenital tract and through the anus. Incubation period. What do we mean by incubation period? Incubation period is the time between exposure to the virus and the development of the first system of the disease which is caused by this virus, اللي هي فترة الحضانة. So if we look at this table, we can see that influenza virus, common cold virus, croup, acute respiratory diseases, usually which are caused by common cold viruses and other viruses, respiratory viruses, the incubation period is categorized by being, or characterized by being of short duration. We're talking about from 6 hours to up to 4 to 5 days, usually the incubation period. And an example that we must talk about also is the coronavirus, SARS-CoV-2. It is found that the average time or duration or incubation period is around 5 days. If we look at this second group, the dengue, herpes simplex, we find that the incubation period is around 1 week. If we look at enteroviruses, it's almost 2 weeks. Poliomyelitis, we're talking about 2 to 3 weeks. When we talk about measles, it's around 2 weeks. Chickenpox, mumps, rubella, all these viruses, enteroviruses, polio, measles, chickenpox, mumps, rubella, we're talking about 2 to 3 weeks of incubation period. So if a child becomes exposed to a child who has mumps or rubella, chickenpox, and after 1 week he did not develop any symptoms or illness, that does not mean he has not got the infection. We must wait up to 2 weeks and to tell if he has got the infection and to develop symptoms. Infectious mononucleosis, we're talking about 1 to 2 months incubation period. We have hepatitis viruses. Hepatitis A, we're talking from 15 to 40 days. Hepatitis B from 2 to 5 months, the incubation period. Rabies virus. Rabies virus is characterized by long incubation period from 15 days, 2 weeks, up to 1 year. It's more even than 100 days, up to 1 year, the rabies virus. And this depends on the multiple factors. One of them is the site of rabies, comes from the bite or scratch from animals which are infected by the rabies virus. Rabies is not a human virus, it's rather an animal virus. Good example of animals, we have the dog, we have the cats can transmit rabies, but mostly dogs. Wild animals such as raccoons, skunks, all these animals might be infected with the rabies virus. And the factors that decide the incubation period are multiple. Among them is the immune status of the virus and the location of the bite of the animal. The closer the bite to the central nervous system, the shorter the incubation period. The further the bite from the central nervous system, the longer the incubation period. So if someone gets bitten in the leg, is not like someone who gets bitten in the neck. In the neck, it's going to have shorter incubation period, usually less than 1 month. In the leg, it might take months. Interference should be during this incubation period. Any person who becomes bitten by a rabid animal should get the vaccine. And this is the only and best thing to provide to this patient in order to prevent the virus reaching the central nervous system. Once the virus has reached the central nervous system, the ultimate fate of this patient is death. We have the papillomavirus, human papillomavirus. Human papillomavirus is known to cause cervical cancer in women, and the incubation period is usually from 2 months to 6 months. HIV virus. The HIV virus also is characterized by a long incubation period from 1 to 10 years. Communicability, another terminology. We're still talking about terminology. Communicability, ability of the virus to shed into the secretions. Communicability is the ability of the virus to shed into secretions. So let's talk about respiratory virus. Once the human gets infected with, let's say, influenza virus. Influenza virus is going to infect the upper respiratory tract. It's going to replicate there. It's going to produce new viruses, and these new viruses are going to be shed into the secretions. And they are going to become communicable. So any person who gets into reach with someone who's infected with influenza, who has coughing, sneezing, and they get into close contact with each other, then the disease is going to become communicable, or is going to be contagious, and it's going to transmit the infection to the other person. Also, another example, good example, is the SARS-CoV-2. Localized infection, disseminated infection. The infection with viruses might be a localized infection or disseminated infection. Localized infection, which is infection limited to the site of entry. Once again, if we talk about influenza virus is a good example of a localized infection. Other upper respiratory tract infection or common cold viruses such as the rhinovirus, the coronavirus are good examples of localized infection. So in localized infection, the virus enters through the respiratory tract, through aerosols or droplets, then it infects the upper respiratory tract. The virus remains there, causes symptoms there, and then the illness start to fade away, and the viral shedding also start to fade away, and the patient is going to recover within 5 to 7 days. It does not go to the blood, it does not go to other organs in the body. That's localized infection. Disseminated infection is the contrary to localized infection. Disseminated infection, it spreads throughout the body. So it starts at a certain location, whether the upper respiratory tract or the GI tract. Then it goes to the regional lymph nodes, and then through the lymph nodes it goes to the blood. And after going to the blood, it might go once again to other organ, infect that organ, and then it is released into the blood once again. So this leads us to the primary viremia and secondary viremia. In the primary viremia, the virus infects at the site of entry, then it goes to the regional lymph nodes, and from the regional lymph nodes it's going to spread to the blood, and this is called primary viremia. If the virus goes from the blood to infect other organ, such as the liver, spleen, kidney, CNS, or whatever, then this is called secondary viremia. And certain viruses are going to spread into these other organs, internal organs, and produce a virus once again, which is going to be released into the blood. This is secondary viremia. So primary replication, having gained entry into a potential host, the virus must initiate an infection by entering a susceptible cell. This frequently determines whether the infection will remain localized at the site of entry or spread to become a systemic infection. When we say primary infection or primary replication, that the virus has entered for the first time into the host and it has infected the certain type of tissues or cells. Once again, respiratory viruses infect mostly upper respiratory tract infection, and primary replication will remain either localized or spread. So primary replication or primary infection is the fact that this virus has entered for the first time, and it depends on the type of virus. The infection might remain localized or it might become a systemic infection. Secondary replication occurs mostly in the systemic infections, in disseminated infections, not in localized infections. It occurs in systemic infection when a virus reaches other tissues, so it infects at the primary site of infection, then spread via the lymph nodes to the blood, and from the blood it goes to infect other organs. Example, poliovirus. Poliovirus, it starts in the GI tract through the fecal oral route, and then through the lymph nodes to the blood. From the blood it goes to the neurons in the brain and the spinal cord and replicate there. So this is secondary replication or disseminated infection. Also we have the lentiviruses, which are the Retroviridae family. Lentiviruses, here we're talking about the HIV, is a good example of the lentivirus, where they spread through macrophages and the CNS to other tissues as well. If a virus can be prevented from reaching tissues where secondary replication can occur, generally no disease can result. This is a good piece of information, where if we can neutralize the virus or get rid of the virus before it reaches the circulation and goes to these organs, then we can prevent these viruses from causing illness. So this is an example of a localized infection and a systemic infection. We have viruses such as the rhinovirus. Rhinovirus is the common cold virus. الفيروس يسبب رشوحات. It usually infects the upper respiratory tract. We have the rotavirus. We talked previously about the rotavirus, which belongs to the Retroviridae family. The rotavirus is associated with gastroenteritis, diarrhea, vomiting, especially in infants. And as we said, it's necessary or important to provide these infants and children with good hydration in order to prevent death with this virus. The primary replication occurs in the GI tract, so this fecal oral. Also, we have the papilloma viruses. Also, in the primary replication occurs in the epidermis, and they are associated with the warts أو الثقاليل اللي ممكن نشوفها on the skin. Systemic infections, we have two examples. We have the enterovirus, and when we talk about systemic infections, we have primary replication and we have secondary replication. Primary replication occurs in the site of entry of the virus and then spreads via lymph nodes, blood to the site of other organs, which secondary replication occurs at. Enteroviruses, they are transmitted via the fecal-oral route through the GI tract, and then they reach the lymphoid tissue and the central nervous system. Herpes viruses, they usually infect through the oropharynx or the genitourinary tract, and then they reach the lymphoid cells and the central nervous system. So they have primary replication and secondary replication, and they are systemic or disseminated infections. Spread throughout the host, apart from direct cell-to-cell contact can occur, and through which the virus can spread to the nearby cells. Also, the spread can occur through the bloodstream or can occur through the nervous system. Via the bloodstream, virus might get into the bloodstream by direct inoculation, such as arthropod vector, blood transfusion, or IV drug abuse and sharing needles. Then the virus is going to travel free in the plasma or blood, such as in the toga virus and enteroviruses, or in association with the red blood cells, such as in the orbivirus, platelets, such as in the herpes simplex virus, or the lymphocytes, such as in Epstein-Barr virus, cytomegalovirus, or even in the monocytes, such as the HIV virus. Primary viremia usually proceeds and is necessary for spread to the bloodstream, followed by more generalized high-titer secondary viremia as the virus reaches other target tissues or replicate directly in the blood cells. So once the virus enters through enters at the site of entry, it's going to replicate there, going to reach the blood, and this is primary viremia. Then it's going to reach the other organs where it's going to replicate there. The titer or the amount of the virus which is going to be released into the blood, that's what we call high titer in the secondary viremia, is going to occur. Also, spread can occur via the nervous system. Spread to the nervous system is preceded by primary viremia. So primary viremia, the virus is going to enter into the site of entry to reach the lymph nodes, and then it's going to move to the blood, and this is what we call primary viremia. In some cases, spread occur directly by contact with neurons at the primary site of infection. In other cases, via the bloodstream. A spread can occur directly by contact with the neurons at the primary site of infection. A good example of that is the rabies virus infection, because once someone gets bitten by a rabid dog, the virus is going to be present within the muscular tissue and the surrounding peripheral nerves there, and it's going to survive and replicate there, and then the virus is going to go into the peripheral nerves, and then it's going to move retrograde. That means from the peripheral nerves moving centrally until it reaches the central nervous system, or in other cases via the bloodstream. Once in peripheral nerves, the virus can spread to the CNS by axonal transport along neurons, such as the herpes simplex virus. The herpes simplex virus, which belongs to the herpes viridae. The herpes viridae family has multiple members. We have in the herpes viridae family 8 members of the herpes viridae family. We have the simplest thing to say about naming those members is the human herpes virus 1 to 8. Human herpes virus 1 to 8. But we have more specific nomenclature for these viruses. So human herpes virus 1 is herpes simplex virus 1. A human herpes virus 2 is herpes simplex virus 2. A human herpes virus 3 is not herpes simplex virus 3. It is varicella zoster virus, the virus which is responsible for both chickenpox and shingles. A human herpes virus 4 is Epstein-Barr virus. Epstein-Barr virus causes infectious mononucleosis or kissing disease. We have also cytomegalovirus, which is a human herpes virus 5, human herpes virus 5, cytomegalovirus, which is seen mostly among immunocompromised or those with chronic illnesses. Also, we have human herpes virus 6 and 7. They are human herpes virus 6 and 7. Human herpes virus 6 and 7 cause what is called Roseola infantum or exantheme sabatum in children illness, mostly seen with human herpes virus 6. Human herpes virus 7 is associated also with respiratory tract illnesses, fever, diarrhea, vomiting as well. And the last member of the herpes viridae family is the human herpes virus 8 or Kaposi sarcoma associated virus, which is seen mostly in patients with skin lesion, blackish discoloration of the skin in AIDS patient. So the herpes simplex virus, herpes simplex virus 1, varicella zoster virus, these are characterized by being latent viruses. And we're going to get into what do we mean by latent virus. So those viruses, they infect and they never leave the body, but rather they go and hide within the central nervous, hide within the ganglia of the central nervous system, and they remain dormant or silent there. And once reactivated, they can spread via axonal transport along neurons. Viruses can cross synaptic junctions since these frequently contain virus receptors, allowing the virus to jump from one cell to another through the central nervous system. Once again, we have some terminology that we need to be familiar with. We have the virulence, is the ability of the virus to cause disease in infected cell. So the virus has infected a certain cell. If it is a virulent virus, then it is going to cause disease in this infected cell. Is there a chance of a virus infecting a cell without causing disease? Yes. This is non-virulent virus. We have persistent infection. What do we mean by persistent infection? In the persistent infection, the infection lasts for a longer period of time, and this is contrary to what we have is the acute infection. So acute infection is infection which lasts for a short period of time. We're talking 5 to 7 days. Good example is the influenza or upper respiratory tract infection caused by the common cold viruses. And we have the persistent infection, which in this case we might have either the chronic infection, which might last for years, such as the hepatitis B, hepatitis C, or HIV, or we have the latent infection. The latent infection is characterized by an acute phase of infection. So the virus enters into the body, it infects, and during this acute phase of infection, the patient might be symptomatic or asymptomatic. If the patient is asymptomatic, does not develop symptoms, then we call it subclinical infection. And in latent infection, mostly the primary infection or the infection for the first time with a virus is subclinical. Then the virus does not leave the body, but rather the virus remains within the body and goes and hides. As we said, herpes simplex virus 1, herpes simplex virus 2, varicella zoster virus, they go and hide in the dorsal nerve ganglia. So how do we characterize the virus by being latent virus. The virus is going to hide in which form in the dorsal nerve ganglia? It's going to hide in the form of an episome. It's going to hide in the form of an episome. Episome is a circular piece of DNA. And the virus is going to this episome is going to contain either the whole genome of the virus or part of the genome of the virus, mostly the whole genome of the virus in the what we call the episome. This episome is going to be in the dorsal nerve ganglia, and the virus is not going to be able to produce the viral proteins. It's going to produce only the early proteins only. It's not going to be able to produce the structural proteins, nor it's going to be able to replicate its genome. That's why it's going to remain silent or dormant there. In the case of any immunosuppression, then these viruses are going to reactivate and they are going to infect humans once again. Most of the infections which occur by the herpes simplex virus 1, herpes simplex virus 1 causes infections which are above the waist. Herpes simplex virus 2 cause infections below the waist, and herpes simplex virus usually cause circumoral infections, either around the lips or on the lips themselves. And usually once this lesion is seen there, this is a result of secondary infection or reactivation of the virus, because we said that primary infection or infection for the first time is usually subclinical. What are the triggering factors for reactivation of these viruses? Stress, infection, exposure to UV light. All these might lead to reactivation of the virus in latent infection. Also, we have another term which is permissive cells. Permissive cells and non-permissive cells. Permissive cells which allow production of virus and or transformation. Non-permissive cells permit cell transformation only. So when we talk about permissive and non-permissive cells, we get back into the virulence. As we said, the ability of the virus to cause disease in infected cell. If the virus goes to the infected cell and the cell can harbor this virus and provide the replication machine or machinery for the virus to replicate, produce new viruses, then this is permissive cell. If the virus goes to this cell and the cell cannot or does not allow the virus to replicate within and produce new viruses, then this is a non-permissive cell. And in this case, the virus is going to be able to produce only the early proteins of the virus, and they might persist and remain within the cell, and later on, it might lead to transformation of the cell. While in the case of permissive cells, the virus is going to replicate, produce new viruses, and maybe infecting nearby cells. And in certain instances, it could lead to transformation of the cell. Transformation, that means from the normal cell to abnormal cell or cancerous transformation. Virulent viruses versus virulent viruses kill target cell and cause disease, which is productive response. When we say productive response, there is production of new viruses. So in permissive cells, infection in permissive cells are going to lead to production of new viruses, and these viruses are virulent viruses, which ultimately is going to lead to death of the target cell and cause disease and spread of the virus to nearby cells. In the case of abortive infection, there is no virus replication. Then if the virus infects non-permissive cell, there is no virus replication going to occur, and only the early viral proteins are going to be produced, and this is going to cause cell death in certain instances. This is abortive infection. So infection within non-permissive cell is called abortive infection. Cytopathic effect. What do we mean by cytopathic effect? Cytopathic effects are the effects of the virus entry into the target cell. So if the virus infects a target cell, then there are consequences and changes that the virus might the viral infection might lead to changes within the target cell. These changes that occur within the target cell are called cytopathic effects. Cytopathic effects. So once the virus enter into the target cell and start replication, once the virus enter into the target cell, the general rule is the virus is going to occupy the cell, it's going to control the cell and control the cell resources for its own benefit. So it's going to colonize the cell and control its own resources for the benefit of the cell. It's going to lead to change in the size and shape in certain instances, cytoplasmic inclusion bodies, nuclear inclusion bodies, cell fuse to form multinucleated giant cells. It's going to lead to cell lysis, might change the DNA, it might transform the cell into cancerous cell. And the last one is virokines and viroreceptors, which is seen in DNA viruses, and cell proliferate and avoid host defenses. Virokines and viroreceptors, we're going to talk in some detail about those 7 cytopathic effects, but it's your duties to go and look up virokines and viroreceptors. They only affect DNA viruses, and how do they affect DNA viruses? And this might come as a question in the exam. So it might change the size and shape. That's clear. Needs no more elaboration. But we have cytoplasmic inclusion and nuclear inclusion bodies. What do we mean by inclusion bodies? Inclusion bodies are viral proteins. Viral proteins are divided into either early protein, non-structural protein, or structural protein. So what do you think inclusion bodies are? Are they structural or non-structural? No, they are structural proteins. So what are the structural proteins? We have either the capsid or the spike. What are the inclusion bodies? Are they the capsid or the spike? They are the capsid proteins of the virus, which are left behind in either the cytoplasm or in the nucleus of the cell in which the virus has replicated and being released. So how was this capsid left in the cell? So virus replication in general is not 100% effective mechanism. So if the viruses produce structural proteins and enzymes, they use, for example, for a certain virus, it might use up to 60% of the produced capsid protein. Example, herpes simplex. They might replicate, produce capsid protein, and not all produced capsid protein is going to be included in new virions. Rather, some are going to be left behind, and these are the inclusion bodies. So what decides if these are cytoplasmic or nuclear? As we said, all DNA viruses replicate in the nucleus except for pox virus. So when we talk about DNA viruses, then mostly inclusion bodies in the DNA viruses are going to be found within the nucleus, except for the pox virus, where we should think of inclusion bodies in the cytoplasm. The other rule is all DNA or RNA viruses, all RNA viruses replicate in the cytoplasm except for HIV and influenza virus. So for RNA viruses, we expect to see the inclusion bodies in the cytoplasm. Number 4, cells fuse to form multinucleated giant cells. or another name for it is syncytia formation. Syncytia formation. Where do we see this multinucleated giant cells or syncytia? We see them in enveloped viruses who enter the cell via fusion mechanism. So let's say the HIV virus. In the HIV virus, we see multinucleated giant cell or syncytia formation. What happens there? So as we said, in the HIV virus, it's an envelope virus, and within the envelope, we have the spikes or glycoproteins. In order for the virus to enter into the cell, the spikes or glycoproteins needs to attach to receptors on target cell. Once these glycoproteins attach to receptor on target cell, they are going to allow for the virus entry into the cell. And the envelope of the virus is going to be left as part of the cell membrane. It's going to be left as part of the cell membrane. Okay? So how does these multinucleated giant cells or syncytia form? Once the envelope of the virus is left as part of the cell membrane, it's going to have the spikes or glycoproteins. And this cell has a nearby cell which is not infected with the virus and which has the receptor for the virus. So the glycoprotein on the envelope, which came part of the cellular membrane, are going to fuse to the receptor on the nearby cell, and it's going to lead to fusion of the cells once again, similar to what occurs in the viral entry. And this is going to lead to having two cells fused together in a big cell with two nuclei or even multiple nuclei. So this is how this occurs. Cell lysis. As we said, in replication of naked viruses, viruses continue to replicate inside the cell, and once the cell cannot cope with the presence of the virus and became exhausted, then it is lysed and released. It's lysed and it dies and it releases the viruses which are inside. It can change the DNA. It can change the DNA either via integrating or incorporating its genome into the DNA, or viruses can produce toxic proteins or enzymes which can lead to changes in the DNA. Transform cells into cancerous cells. When we talk about transforming cells into cancerous cells, we're talking about DNA viruses mostly, but also certain RNA viruses might lead to cancerous transformations. And cancerous transformation, as you remember when we talked about the replication, when we talked about the replication in DNA viruses, and there are some obstacles that can occur. One of them is the S phase thing. Do viruses wait for the S phase, or do they oblige the cell to enter into the S phase? And we said that most viruses oblige the cell to enter into an S phase, continuous phase of replication. So this is one mechanism of virus transformation. Number 8, remember, this is your duty to look. Cytopathic changes. Here we see multiple nuclei within a single cell or syncytia formation. As we said, this occurs in envelope viruses, and we've already talked about the mechanism or how does this occur. Here what we see is inclusion bodies, and we said inclusion bodies are remnants of the capsid protein of the virus, which can be left in the nucleus or the cytoplasm, depending on the site of replication of the virus. These are examples of cytopathic changes that might occur in the small box. The cells round up and inclusions appear in the cytoplasm. Smallpox belongs to the poxviridae family, which is a DNA virus which replicates in the cytoplasm. So that's why we see cytoplasmic inclusion bodies. Herpes simplex cells fuse to form multinucleated giant cell, and we might see nuclear inclusion. Adenovirus, there is a nuclear inclusion and clumping of the cell. Poliovirus, no inclusion and leads to cell lysis. The poliovirus is a naked virus. Reovirus, cell enlargement, vacuolation, and inclusion in the cytoplasm. Reovirus example is the rotavirus. Influenza virus, cells round up, no inclusion. Rabies virus, no change in the cell shape, but rather we see cytoplasmic inclusion. And these are called Negri bodies, and they are diagnostic of rabies virus infection. The rabies virus is an RNA virus which replicates in the cytoplasm, and that's why we see the cytoplasmic inclusion. The HIV is characterized by multinucleated giant cell formation. So here we have the HIV, we have the herpes simplex virus, among other viruses which might lead to the formation of multinucleated giant cell and syncytia formation. Go look for other viruses which cause syncytia formation, and this might come as a question in the exam. Patterns of viral infection. We have the inapparent or subclinical viral infection when human becomes infected with the virus, but he does not really know or develop any symptoms as a result of infection, but rather he develops immunity against this infecting virus. We have apparent infection اللي هو ظاهر هيكون عندنا infection ظاهر, and in this case we have the acute infection, and a good example is the common cold or the influenza virus infection, where you develop the cold symptoms, cough, sneezing, headache, fever, all these, and it lasts for 5 to 7 days and then complete recovery. That's acute infection. Persistent infection, which lasts for a longer period of time and is divided into chronic infection, latent infection, and slow chronic virus infection, and we're going to talk about these in a little bit of detail in a moment. So these are examples of pattern of viral infection. Here we look at these figures, and just to know, this red pattern represents the symptoms which develop in the patient, and this blue line represents the virus production, peak of virus production, and then the disappearance of the virus. This is acute virus infection. In the acute virus infection, the virus enters into the human body, it replicates, and from here to here, this is the incubation period. So the virus enters here, this is the incubation period until the development of the symptoms here. So these are the development of the symptoms or the illness, and then a couple of days the virus start to fade away, and or the production of the virus slows down, and the immune system also helps in clearing the virus and getting rid of the virus until the virus is being cleared totally from the body within 5 to 7 days. Good example is the rhinovirus, the rotavirus, the influenza virus. Here we have the persistent viral infections or the chronic infection. Chronic infection. Example is hepatitis B and hepatitis C. So the virus enters here, and this viral infection might be associated with acute phase of infection. So the patient might develop symptoms here, but mostly when we talk about hepatitis B and C, the patient does not know that he has been infected. Subclinical infection, primary infection is subclinical infection. the virus titer, or the amount of the virus in the body, starts to rise until it plateaus, youthput, and once the virus infection is advanced, after years of the virus infection and damage to the liver cells, the person starts to develop symptoms, and during this period, the immune system is in a battle with the virus in the blood, and there are some ups and downs. It's not really straight, some ups and downs in the amount or the titer of the virus, and the ultimate fate of those patients is death after a long period of time, because the viruses in the hepatitis B and C infect the liver cells, and at the same time, let's say the 40 or 50% of the liver cells are infected and the other are healthy. But once the infected cells are going up to ultimately die, and we know that the liver is capable of regeneration, so those cells are going to die, regenerate. Other healthy cells are going to become infected. They're going to die, regenerate. This round of infection regeneration is going to lead to fibrosis of the liver cells. Then it's going to lead to cancerous transformation of the liver cell, hepatocellular carcinoma, and ultimately the patient is going to die. So this is chronic infection. This is latent reactivating infection. Latency, we talked about the latent virus, and a very good example is the herpes simplex viruses. And we said that in latency we have an episome, production of the early enzyme only, and they seek refuge in the dorsal nerve ganglia for herpes simplex virus 1 and herpes simplex virus 2 and varicella zoster, and they seek refuge at other locations for other herpes viridae members. The virus, the primary infection is usually asymptomatic. The patient do not develop symptoms, and then they go and hide, and then reactivation occurs. So this is a reactivation where the virus reactivates, travel from the dorsal nerve ganglia into the skin and cause lesion. Herpes simplex virus 1, we said they are circumoral, and then the immune system kicks in, decrease the amount of virus, and leads to clearance of the virus, but not clearance as in here. The virus is going to go here into the dorsal nerve ganglia once again, and it is associated with reactivation, as we said, upon when the triggering factors such as the exposure to sunlight, UV, infection, stress, trauma occurs, and there is suppression in immunity, reactivation might occur. Reactivation might be symptomatic or asymptomatic. Also, we have acute followed by persistent infection and virus overproduction, and a good example is the HIV virus. So the HIV virus is going to infect a human in here incubation period, which is around 4 to 6 months. Then the patient might develop symptoms in the acute phase of the illness, which is a strong influenza-like infection. Then the virus amount or the immune system is going to kick in, and it's going to lead to reduction in the virus titer in the body. It will never go back to zero. The virus is going to remain within the body. It's going to be replicating all the time during the body, and as we see, these peaks develop when the immune system goes down, the viral titer increases, and once the immune system is stronger and more robust, the viral titer is going to go down, and this is going to take years, 10, 20, 30 years. And now with the presence of the antiviral drugs against the HIV virus, the persons might die of natural causes rather than dying of the effect of the HIV virus. Once we reach to this stage, the patient is characterized as having AIDS, autoimmune deficiency syndrome. And we need to differentiate between HIV infection and the development of AIDS. So in AIDS, the immune system of the patient has been almost destroyed and the CD4 count is below 200. During these periods or these years, the CD4 count is more than 200 and the amount and the immune system is still capable of doing its job partially. It's in battle with the virus in the blood, but here we can see that the virus is winning. Here the immune system is winning the virus, the immune system, until we reach a point here where the immune system almost collapses. CD4 count goes below 200, and then the patient has the symptoms characteristic of AIDS. We have also slow chronic infection, and in slow chronic infection, viruses such as the JC virus might be present, JC polyomavirus. The virus infects here and is not being recognized unless assayed for, and it takes years in order to be detected or develop any symptoms. So this is chronic infection. Once again, hepatitis B, latent infection, herpes viridae, slow virus infection such as the JC polyomavirus or the progressive multifocal leukoencephalopathy. Here we have the virus cell interactions which might occur in vivo. In the acute infection, it is characterized by being cytocidal, and cytocidal means that the ultimate fate of the cell infected with the virus is death. That's why we say cytocidal. In the acute infection, is there cell death? Yes. Is there infectious virus production? Yes. And we can see that the virus is being produced. The cell ultimately will die and it's going to infect nearby cells. Example, influenza, polio, toga, encephalitis. Persistent viruses, we have the chronic diffuse infection. Is there cell death? No. Is there infectious virus? Yes. And in this case, the cells continue for long period of time. We're talking about months or even couple of years producing viruses without being targeted by the immune system, because the immune system is being weakened by these viruses and it's no longer performing how it should be. And these are seen in congenital rubella and congenital lymphocytic choriomeningitis. Congenital, where we mean that they are transmitted vertically from mother to baby, and during pregnancy they cause congenital rubella or congenital lymphocytic choriomeningitis, and they are associated, if they occur within the pregnancy, they are associated with deformities or malfunctions within the immune system, and that's why they remain producing. But the rubella itself is characterized as being acute. The congenital is being chronic diffuse, where there is infection of the virus but no cell death. A chronic focal, a fraction of the infected cells are few. Is there cell death? Yes, there is cell death. Is there infectious virus? Yes, there is infectious virus. A good example is the adenovirus infection within the adenoids. I'm not going to elaborate more on this. Maybe we'll talk about it later on, but the adenoids might become infected with the adenovirus. The virus might be produced, lead to cell death, and later on also it might lie dormant within the adenoids, and is cleared by the immune system. In latent virus infection, we do not have cell death. we do not have production of the virus unless there is reactivation of the virus, as we said in herpes simplex virus 1 in the dorsal nerve ganglia. And a good example is the herpes simplex virus 1, 2, or even the varicella zoster virus. So the overall fate of the cell die in the cytocidal infection, and this may be acute or chronic, only a few cells infected while the rest proliferate. This is cytocidal effect. The cell lives in persistent infection. This may be productive or non-productive. Productive, where the virus is capable of replicating and producing new viruses. Non-productive, we said when the virus enters into the wrong cell, is going to produce only the early proteins, and it might lead to transformation of the cell. And we have also the latency, as we have talked about in herpes simplex viruses. We have transformation. Transformation is the cells transform from normal cells into cancerous cells, or the viruses might lead to tumor production. RNA tumor viruses usually transform cell into a malignant phenotype by integrating their own genetic material into the cellular genome, and may also produce infectious progeny. And here we're talking specifically about HIV. So HIV belongs to the retroviruses, and the retroviruses, how do they lead to transformation of the cell? Either through acute transforming virus. In the case of acute transforming virus, we have the viral SRC gene, and when we say the viral V, then we have the C cellular SRC gene. So those genes are present both in viruses and in humans, and it was thought that these viruses were human viruses originally, and when the viruses has infected years and years, hundreds and thousands years ago, they have acquired these genes from humans, and they became as part of their genome, the viruses. So we have the viral SRC and we have the cellular SRC. In the acute transforming viruses, the viral SRC oncogene mimic cellular genes, and that's what we call proto-oncogene. So this is a viral gene, and it is being, once the virus infects the cell, it is, as we said, it's going to control the cell, produce its own proteins. So once the VSRC is produced within the cell has the CRC, the cellular gene. So overproduction of the viral is going to interfere with the cellular pathways of the host cell, and is going to transform the cell into a cancerous cell. Also, we have insertional mutagenesis, inappropriate expression of a proto-oncogene adjacent to the integrated viral genome. In this case, what occurs is once the virus enters into the cell, the virus is going to integrate its genome into the human cell. It's going to integrate its genome into the human cell. So, in order for a protein to be expressed, it has to be preceded by a promoter. It has to be preceded by promoter. So in the case of insertional mutagenesis, what occurs is inappropriate expression of the proto-oncogene adjacent to integrated viral genome. So if the cellular promoter, or let's say if the viral gene is inserted in front of the promoter, it's going to lead to expression of excess of the proto-oncogene, and it's going to interfere with the cellular pathway of the cell, and it's going to lead to transformation. Another thing is the transactivating factors. Certain genes within the HIV or the HTLV in the retroviruses are toxic to the cell, and they turn the cell gene, they turn on the cellular genes, causing uncontrolled cellular proliferation, such as the TAX gene in the HTLV. is going to lead to continuous proliferation of the cell, and it's going to cause a transformation of the cell. Also, we have DNA tumor viruses are often cytocidal. DNA tumor viruses are often cytocidal, and they lead to death of the cell ultimately. But some of these DNA infections might lead to an abortive infection or restrictive infection, might be associated with the production only of the early genes of the virus, and they might lead to transformation of the cell. Also, we have the human papillomavirus might interfere with p53 or retinoblastoma genes, which regulate the cell cycle. p53 function as a tumor suppressor that's involved in the preventing cancer. A human papillomavirus interfere with the p53. Also, human papillomavirus interfere with retinoblastoma, which prevents excessive cell growth by inhibiting cell cycle progression until cell is ready to divide. And by interfering with this mechanism, human papillomavirus might lead to cancerous transformation and development of cervical cancer. Also, the cells might enter apoptosis, and apoptosis is programmed cell death. Once the human cell, is incapable of replicating and doing its function properly, then it might initiate self-apoptosis or self-death of the cell. But p53 initiate apoptosis, programmed cell death, if DNA damage proves to be irreparable. If viruses interfere with the p53, they are going to interfere with this programmed cell death, so the aging cell is going to divide in an erratic manner and is going to lead to cancerous transformation. Types of viral infection at the cellular level. We have the abortive viral infection, virus production. There is no virus production, fate of the cell, mostly nothing, or it might lead to transformation. Cytolytic or cytocidal, the virus is being produced and the ultimate fate of the cell is death, اللي هي بيصير في عنا في الacute infections. في persistent infections في عنا persistent productive infections where there is a virus produced, and this is going to lead to senescence of the cell. In the latent virus infection, there is no virus production and the cell is not going to be affected. Senescence here means holding the cell steady in a state of non-replication. Transforming viruses, we have DNA viruses. Are they associated with virus production? No, we said DNA viruses. They might infect the cell in an abortive infection, produce only the early proteins only, and they might lead to transformation. There is no virus replication. The RNA viruses, there is virus production, and it is associated with immortalization, as we said in the HIV. Mechanism of viral cytopathogenesis, inhibition of the cellular protein, inhibition and degradation of the cellular DNA, such as in the herpes virus, alteration of the cell membrane structure in all enveloped viruses, and toxicity of virion components, such as in the adenovirus fibers. So the possible consequences to a cell that's infected by a virus are lytic infection, persistent infection, latent infection, and transformation, as we have talked in detail in this lecture. Once again, this is a diagram to show the fate of the cell that can occur as a result of a virus infection. That's all for today. Thank you for listening, and shortly we will schedule a live session. If you have any questions about the first 3 lectures, I'm happy to answer any of these questions, so please be prepared with all your questions, and it will be announced soon, inshallah. Thank you.